Okay, great. Thanks, Matt. Oh, that's your phone, not mine. So it's great to be here. It's wonderful to be kicking off this 20th EEID. Matt asked me to reflect on 20 years of the study of disease transmission and how the field had moved on over that time. That's a very big and difficult question, and we spent a lot of time thinking about titles, and seems that basically transmission is maybe not everywhere, but it's everything, and it's definitely not all at once. I talked at the first EEID. I was a child prodigy. I was very young at the time. And I think I've been to all but two over that time. One I had no excuse for missing, and one my son was really inconveniently late joining us, so I had to miss one. And I think I've only talked at one other EID as far as I can remember, but I've gone to all of them. And I don't do that with any other conferences. And in this conference, I think it's been incredibly influential on my career and many of the careers of the people in this room. So I think maybe the title should maybe be like 20 years of EID is everything, everybody, everywhere, but not all at once. Now, the first EID, as people have said, was very small. That's, I think, all of us on a deck. And it's Pete Hudson's deck, so it's a Pennsylvanian deck. So as decks go, it's a big deck. But it was hardly, very, very few people coming together really to, as Matt said, build community. And that's developed into this much larger, mature, more diverse EEID community. So, and I think this community has had a huge impact in dealing with the, I'm going to call it the current pandemic, it's not over. So what I thought I would do was also try and reflect a little bit on the role of EEID and the thinking that's kind of developed from this community that came out of this first meeting, and how that's really impacted the response to COVID-19. So what I'm going to do is quickly, I'll revisit the theme of my talk 20 years ago, the evolution of virulence, and talk a bit about show you what I presented then, and talk a little bit about how that field has developed, and present some new work as well. So there's some science in this talk on some sort of cutting edge work on evolution of virulence that we're doing now. But I also want to talk a little bit about the impact of EEID, and also what is it about EEID? What is it about ecology, evolution of infectious disease that sets it apart? Of course, everything's collaborative. I do very little myself now, and the model I'm going to talk about was developed by Kirzazaki and Sebly, and working with those guys. Okay, so let's start with evolution of virulence. So as many of you know, evolution of virulence work really is based on this idea that there's a trade-off, the trade-off theory of the evolution of virulence, which is predicated on the idea that parasite traits like transmission and virulence may trade off against each other, so that there's some sort of optimal fitness for any particular pathogen. This is a slide I stole from Jaap de Rood many years ago. And the basic idea is, if you think about very simple within host model, if you like, of host parasites, you have parasite replication. If parasite replication's higher, you can get more transmission stages. But the idea is that the trade-off is that with those more rapid replication, you're going to do more damage to the host. There will be more mortality, which is virulence, shortening infectious period, and therefore the costs and benefits of transmission and mortality end up leading to some sort of optimal fitness, optimal parasite replication, optimal transmission, optimal mortality. So I talked about this 20 years ago. One of the key things I think that's changed is the data that we have that's supporting these ideas. So 20 years ago, we'd talk a little bit about myxomatosis and all that original data. And that was the data that kind of inspired May and Anderson to start thinking about trade-offs and the evolution of virulence, although they initially thought about the clearance-virulence trade-off. And then we had some experimental data, notably on rodent malaria systems, but there actually you're often implying the virulence through measures like decreased birth rate. But now we have a whole bunch, over this 20 years, been a whole bunch of beautiful experimental programs in multiple systems that make it very convincing that at least in these systems, there is some sort of optimal level of virulence. This is Jaap de Roos and group's work on monarch butterflies, optimal virulence. In some human diseases as well, Christoph Fraser's work on HIV, optimal virulence, and more recently, the detailed work on Merrick's disease, which kind of has everything, has within growth loads, has shedding, has transmission. So I think we have much better data now that makes sense to think about studying the optimal evolution of virulence and transmission. So fundamentally, the idea, virulence is a cost to transmission. If you have a transmission-virulence relationship, it goes something like this, so that virulent transmission gets increasingly costly. You end up at some sort of optimal transmission and virulence. And in simple models with equilibrium population dynamics, mixed mean field models, then that will often be the transmission virulence on this trade-off that maximizes R0 for the parasite. Okay, so what was I interested in 20 years ago? 20 years ago, we were working on spatial structure and the evolution of virulence. So we were trying to understand how, if you put a little bit of spatial structure, population structure into a model, so that you don't infect everybody equally, but you tend to infect people close to you a little bit more, what does that do to the evolution of virulence? These are results that I presented there. And the main results were that if you add local interactions, so here you have, there can be a local interactions and infection from an infected individual next to a susceptible individual, and you can have global infection as well. And if you look at what the optimal virulence is in the model, you can see that when everything's mixed, so infections happen completely globally, you have higher virulence, higher transmission, more acute parasites, and local interactions tend to select for more prudent, lower transmitting chronic parasites. So there's a bunch of other results that you can find in spatial models, one of which is, so I also talked about 20 years ago, is the fact that you often can get these bistabilities in spatial models where you can get positive frequency dependent, positive selection going on, positive frequency selection going on, such that you can get sudden shifts from low virulence to high virulence, just again caused by this structure. Okay. So where's that field now? Well, we've done a lot of models of the role of spatial structure and population structure on evolutionary outcomes. And it matters a lot. I think we have a pretty good and detailed understanding of when spatial structure matters. But also, I think one of the things that's really changed for me when I was reflecting on it is we also have a really good sense and theory of why spatial structure matters and why it's always likely to matter. So if you can assume that your model, your population is very mixed and mean field, then that's fine. But as soon as you think there's some sort of population structure in there, there will be some feedback on the evolutionary process. And the reasons for that is ecological correlation. So if you're transmitting locally, you tend to surround yourself with infected individuals, and that stops you spreading through the population. I call this genetic correlations. This is also inclusive fitness. It's not just when you're spreading locally, you also tend to be particular virus strains are competing for susceptibles with themselves because everything's happening locally. Okay. But the other thing I think that's really important that comes out of these models is that local demography, things like assumptions about reproduction and local death rates, so ecology, the real ecology of the hosts, really starts mattering as soon as you've got some population structure. You can sort of ignore all of that when everyone's mixed together. But these local ecologies, these local demographies can actually be as important as the epidemiological relationships, to be as important as whether the infection is local or global. It also really matters whether infected individuals are reproducing or not. Okay. And that all starts to matter because you have even a little bit of this population structure. And I'll point you to Lewis Bartlett's theoretical paper on this in JTB. He doesn't just do bees, he's also a great theoretician. Okay, so where's evolution of virulence theory now? Well, I think it's an exciting time. I think we're thinking a lot about different constraints on virulence and transmission. David Kennedy gave a great post on that last year at the EID. I think we need to also worry and think about different trade-offs. What actually matters? What actually is important to the fitness of the parasite? What about persistence in the environment? Factors like that. And then the thing that I'm also really excited about at the moment is non-equilibrium dynamics. Until recently, most of this theory just assumes that pathogens, that the host population is constant, and that the what's happened is the pathogens come into the population, it's gone endemic, and it's just sitting there at a steady state. We all know that's really not the way that pathogens are typically. So we've been working on new methods to look at non-equilibrium dynamics and the evolution of virulence. And the first of these I'll tell you a little bit about is antigenic escape. Many people in the audience are familiar with antigenic escapes. Just the idea, if you have a disease, a kind of model disease where there's immunity built up in the population, and then the virus, whatever, mutates away so it can then invade previously immune individuals, and you can end up something like flu with these ladder-shaped outcomes. I'm going to tell you about this again as another pull back to the first EID. This is a paper Julia Gogg and Brian Grenfell. I think Julia presented at the first EID on antigenic escape. And we're going to use this model and then look, this model describes the dynamics of antigenic escape really nicely. And then we're going to use this model and see, does this matter for the evolution of virulence? What I love about this picture is it's Julia talking science with Mary Pass, lots of science conversations like Andy Dobson. Is that Yanis Antonovich? And I'm just sat there with a beer looking off into space. So nothing changes. There you go. Okay. So what's the dynamics? So the simplest idea with antigenic escape is you just get a wave of new parasites coming through all the time. Is that working? Oh, slowly. Okay. So you get this wave, runs through the population, got immunity ahead. That's very boring and slow. Okay. And then the other thing that can happen, maybe a little bit more realistic, is if you assume there's some cross immunity, then the mutations need to build up in order to be able to escape the immunity locally. And then at some point, you suddenly get a new epidemic as you've mutated far enough away that you can take off again. So you get these big outcomes. Repeated epidemics due to immune escape. Okay. Sounds familiar. Okay. So what does this mean? Okay, so if you have these dynamics, either without cross immunity, this wave, or these continual outbreaks with some cross immunity, what the simulations tell you is you get the evolution of much higher virulence. This blue dot, this dashed line here, that's what maximizes R0. That's the level of virulence and transmission that would evolve in a constant population, the classic theory. And just these continual immune escapes select for much higher virulence. If you like that red dot, I'm kind of excited about that. That's an analytical result from a new technique of ligomorphic dynamics some people might be interested in. It allows us to actually predict evolutionary outcomes in non-static, non-equilibrium situations. So I think this is important because what this says is that if you've got a dynamical system where you're getting continual immune escape, you're actually going to select for more acute parasites. And the reason for this is you're continually selecting for high transmission, even if it's costly, because you're continually outbreaking all of the time. Okay, if you're interested, any theoreticians are interested in these techniques, then they're here. All right, that's a new theory. So how about COVID-19? And can this evolution of virulence theory tell us anything about COVID-19? I think when you first were reading Twitter at the start of this whole thing, there were lots of scientists saying it would be evolved to be less virulent. My dean told me that, my molecular biology dean told me that. Of course, this is what's going to happen. I mean, one of the big questions, of course, are the trade-offs? Where are the trade-offs? Do they exist? Are they transmission virulence? Are they transmission immune escape? Looked a little bit like that at the start, then Omicron happened, and then whoop, don't write that paper. Clearance rate, persistence, who knows? Are those trade-offs there? Maybe they are at some point, but maybe not. And then on those points, the important thing is something, if you look at this idea of Pareto fronts, and this is in a little paper we wrote in lockdowns led by Lisa Vischer, who's going to talk tomorrow, I think, or Thursday, maybe. And the point is that, even if there is a transmission virulence trade-off somehow, there's some fundamental mechanistic processes fundamentally that will lead to this in the end. When a disease evolves, it could be anywhere, right? It's going to come into this accessible suboptimal phenotype space, and then you might see lots of what seem to be no-cost mutations as transmission just builds up. So we might be nowhere near any trade-off, but we might get there at some point, and you might end up at some optimal place. But there's no reason to think we'd be close to it at the start. So you might see a lot of non-costly transmission gains. Okay. Non-equilibrium epidemiological dynamics. So I do think these models, like the one I just showed you, are important to try and understand evolutionary dynamics, or at least the selection and selective forces that are happening to something like COVID-19. Because if it keeps outbreaking, I think we will keep getting selection for transmission. And even if the cost is then virulence, it will also be more acute. Okay, so if evolution of virulence is not particularly useful for COVID-19, where's the big evolution EID impacting COVID-19? Well, I think phylogenetic has been really influential. And in this paper written by Brian Grenfell and Eddie Holm and others, this came out, I don't know if it was the first one, maybe the second or third EID, kind of led to them producing this really influential review, which kind of said, you can understand epidemiological processes from the phylogenies, from all this sequencing data that you've got. And that's been, I think, hugely influential and hugely impactful in the pandemic, with a lot of these kind of papers on sort of phylogeographic approaches to working out where the virus came from and the pathways of the virus from the sequence. And I think it's also been used conceptually as well, where it's defined quite a lot of this paper is actually about the conceptual idea of trying to understand processes and predict processes by some sense of knowledge of how diseases evolve. I think that's the big impact from an evolutionary point of view. Of course, the other thing we've all done is we've all built epidemiological models, or the vast majority of us built a COVID model over this time. That's not according to me, Matt. Anyway, all right. So a lot of us have built a COVID model over this time, and not just people in this community. A lot of people have built COVID models, and that's been good and bad. So what did EID and EID thinking bring to that? Well, one of the things I think it probably didn't much is asymptomatic transmission. I don't remember us talking about that at all at an EID, or doing any conceptual stuff or any theoretical stuff around that. I mean, it existed. There's an old paper. It's a great paper by Christoph, which talks about when a disease is uncontrollable. And of course, the main one they have in there is asymptomatic transmission. But of course, we can all build these sorts of models. And we actually built a model on the Berkeley campus because Jennifer Downer converted the Innovative Genomics Institute into a testing center. And as well as providing testing for the Bay, she wanted to see if we could do some asymptomatic testing to get everyone back to Berkeley. Are you sure about the timings, Matt? I've got 20 minutes so far. Okay. Okay. All right. All right. So we're just having an argument. I think you ran on. Okay. So they wanted to start running an asymptomatic testing program on campus to try and convince the chancellor so we could all go back to our labs, basically. Kara Book volunteered. She was doing lots of testing in the labs, I think as many people do, but they asked us if we could build them a model to kind of work out how you should do this. And this is the model. And this is basically measles, SIR, with the addition of pre-symptomatic, asymptomatic. And of course, you need that because the test will take people into isolation at that point. And what did they tell us? As lots of people found, the key thing, of course, is the test turnaround time. So the time between giving someone a test and when you actually tell them the results so that they can isolate. If it's 4 or 5 days, you forget about it. Right? And this was really important. And this was kind of EID dynamical thinking. And when you're talking to molecular biologists and telling them, you don't need to make your test more accurate, that really doesn't matter at all. It's kind of hard for them. They spent their whole life developing accurate things so they can invent gene editing that changes the world. But it didn't matter at all. It just needed to be fast, fast, fast. And of course, we should have had rapid tests at that point, but that influenced the way we were doing that. The other key thing, I think, from these sorts of models this community brings is the whole idea of heterogeneity and superspreading. This is a hugely influential paper by Jamie Lloyd-Smith. And again, you see that classic in a thing that you'll see in our models. If you let all the fat parties happen, then we just can't control it, no matter how much testing we're doing. And very useful things. Okay, so I'm just going to finish quickly by talking about, what can this community do going forward? And what's the ecology and evolution of cross-species transmission? What do we know about this? How much theory do we have on this? Some, not much. How much understanding do we have beyond it just sort of randomly happens and we can't predict it? I think one of the things this community does well is we look at data, we collect all the data we can find, and we look for patterns and we try and understand it that way. This is some work by a student, Sarah, again with Kara. And this, you can look at all the data and you can show quite clearly that bat-borne viral zoonoses are the most virulent of all the zoonoses. They're up here, the purple ones here. But I think something that's nice about this community is we want to go beyond that and actually try and understand why that might be. And we think it's because, well, one of the reasons is bats have these unique physiology, highly tolerant to the disease, weirdly long lifespans for their size. And Kara's producing a model I hope we're out soon, which is a very EID model within host growth, between host growth, and showing how if you have a lot of tolerance in the bats, they have these highly replicating parasites potentially. And then when they jump into humans or other mammals that don't have this same tolerance, it's very bad news. Very quickly. I don't want to have a go at bats. So this is another analysis we did, whereas if you actually look at the death burden of zoonoses that come from different groups, the bats aren't the worst, although up here, that's SARS-CoV-2. If you look at the death burden against the case fatality rates of virulence and look at SARS-CoV-2, it's right down here, but hugely influential. So virulence isn't the key thing most of the time anyway. Okay. So yeah, for cross-species transmission, we need surveys, we need models, we need data fitting, we need inference, but I think we also need theory and model systems. And it's great that there's people like David Kennedy developing model systems, and we're trying to work on honeybee viruses and how they spread out in the population so that we can get a better sense of how these processes really work. Okay, just to conclude then. So I said ecology, dynamics, demography, these are all fundamental to the evolution of infectious disease. And I think that this meeting is where that work happens, that interaction of ecology and evolution. To me, it's ecology and evolution in EAD that makes us distinct from public health or One Health. We overlap with those communities, but we're really interested in ecology and evolution. And at the center of it is there's a lot of conceptual theory we build on, model systems like measles that we build on, and these are our sort of key approaches. And I do think that conceptual theory and model systems are needed to better understand the ecology and evolution of cross-species transmission and emergence. Final. We spent a lot of time trying to come up with the title. And we started with this, who requires infection for human. To me, that's public health, that's EPI, okay? So what did we come up with for EAID? Quite a lot more. So who requires infection from whom, when, where, how long, how often, and at what cost, okay? And that ended up as that, which is too long. And then I decided I didn't want that title tweeted out. And just a shout out to people from my group who are talking today or giving posters at the meeting. So Claire Everson's going to talk about who acquires infection from human at what cost, evolution of virulence. Graham's going to be talking about how, in hyperparasites and at what cost, evolution of virulence. Nina's going to be talking about who acquires infection from human when. And Elisa's going to be talking about who acquires infection from whom and where. And I'll leave you with the conclusions. Thank you.
We have a few minutes for questions. Thank you, Mike, for staying on time. See Sam's hand up.
Yeah, I see. Great. So I think so people started tweeting about the paper about that. So when we had the travel bans and when we had the lockdown, you would be localizing potentially infection, and in principle that could select for lower virulence, lower transmission from that basic theory. But actually the problem is, with people, you don't really have that ecology. You don't have that local birth death processes. And that's actually the thing that drives these results. So we've done some work recently where we're looking at how superspreading affects the evolution of virulence. And what you'll find is that the final optimum doesn't change, but the speeds at which you get there really do matter. So we need to move away a little bit, I think, in humans from what the optimal thing, what the optimum is. And actually, again, it's this transient non-evolution. And then I think it can matter. But I think if anything, it will help a little bit.
Mike, you mentioned in one of your slides the systems, but you didn't actually sort of get to it.
Yeah.
I just wondered if it's about time we stopped and looked at our assumptions that pathogens are always maximizing R naught.
Yeah.
We should consider that in the fact that R naught never maximizing R naught doesn't actually give us persistence. Is it time to review that?
Okay, it's a good question. So yeah, so that's another place I think that evolution of virulence theory can go, which is when you start thinking about what's the stochastic outcome. So if actually what's happening is pathogens are coming out for a while, disappearing. So the classic theory, that's looking at some long-term outcome. And what we haven't done much of, so we know where we're going, but we don't know how we're going to get there, okay? And I think that's where we should start spending a lot of time if we want to try and predict what's going to happen with human disease. But trying to predict what's going to happen with the evolution of human disease, that's exciting, but it might be very difficult. But I think we have to do that. And then that kind of really gets us into a sort of different realm as well. We really need population genetics at that point, because it's really going to be about stochastic processes. And I hope that all this genetic data that we've been getting might allow us to get at that. Yeah, I'm not sure we're going to get very many simple theoretical predictions. I think we need to get to the data a little bit with that and how quickly we get there.